Morning, everyone. Nice to meet you again in this beautiful morning. How are you doing all? Okay, thank you so much. I'm good already. Now, I'll share with you a protein metabolism part 2. The topic of my discussion for this morning will start from urea cycle, amino acid synthesis, amino acid is a source of energy, and the last is a protein turnover. Now let's see urea cycle. Amino acid deamination result in ammonium production. Accumulation of ammonium in human body will cause fatality due to its toxic nature. The liver can convert ammonia and carbon dioxide into urea. And after urea is formed, it can be secreted in the urine. This is about or around 20 to 30 gram daily. Let's see the structure of urea in this figure. See this one. We have two ammonia group in it, and this is ketone group. Urea cycle begin in the mitochondria and will be in a cytoplasm. Free ammonia will be skipped. The reaction use ATP and bicarbonate, producing carboxyl phosphate. Carboxyl phosphate synthesis require energy as activator. Let's see this equation. We have NH4 plus here, and bicarbonate, and this will be converted into carboxyl phosphate by using chemical energy as ATP, and this reaction will be catalyzed by carboxyl phosphate synthesis. Carboxyl phosphate will combine with ornithine, result in another non-protein amino acid, citrulline. Then this citrulline is transferred from a cytochrome to cytoplasm by ornithine translocase. Let's see this reaction. We have in hand carboxyl phosphate, and this carboxyl phosphate will react with ornithine, and this will convert into citrulline. Then the citrulline in mitochondria matrix will be transformed into cytoplasm by ornithine translocase. Now, let's see this one. Citrulline will react with aspartate to produce arginine, and if this arginine is not needed, it can be hydrolyzed into urea and ornithine. See this cycle. Then let's move to fat of urea. Urea formed from the urea cycle will enter the bloodstream. Urea enter the kidney, filtered and extracted in the urine. Let's see, we start from this protein. This protein can be exogenous or endogenous, and this is occur in a liver organ. Then this protein will be break down by proteolysis to produce amino acid, and this amino acid will be converted into acetyl-CoA sometime and other toxic molecule such as ammonia. This ammonia will enter urea cycle to neutralize this ammonia. Then the urea will excreted from a kidney, it's about 90%. Then it will be secreted in the urine. Now let's move to amino acid synthesis. In human, 10 proteogenic amino acid can be synthesized. This is what we call as non-essential amino acid. Essential amino acids are gained from a diet. So let's see this table. This is essential amino acid, and this one is non-essential amino acid. Essential amino acid means that we'll take this one from the diet, but this non-essential amino acid can be synthesized in our body. Now let's move to amino acid synthesis. Amino acids are synthesized from intermediate of major metabolic pathway, such as a pyruvate, oxaloacetate, and ketoglutarate. And transamination reaction of the alpha keto acid is catalyzed by amino transferase. Let's see the equation in this right side. We have two molecules here. Then this molecule will convert into these two new molecules by catalyzing of transaminase. Let's see this one. This is a ketone. Then this ketone will be converted into this molecule. Then this molecule will be converted into a ketone. That's why we call this one as a transaminase. Now let's see one by one amino acid synthesis. Glutamate, glutamine, and proline. These three amino acids share anabolic pathway. Start with glutamate dehydrogenase and alpha ketoglutarate. See this one? Alpha ketoglutarate will convert into a glutamate by oxidizing this one. This is also a chemical energy. Then this glutamate will convert into a glutamine. Glutamate is amino group donor for production of all amino acid. Once again, the glutamate is the amino group donor for production of all amino acid. Then let's see this one. We have this one glutamate. Then this glutamate will be compared into a glutamate hamma semialdehyde. Then this gamma semialdehyde will be compared to delta pyridine 5 carboxylate, and ending a story of this reaction is a protein. Now let's see alanine. Alanine formed through transamination from glutamate and pyruvate. Let's see this one. This is a glutamate and this is a pyruvate. Then these two will be compared into alanine and alpha ketoglutarate. Then how about Asparta and Asparagine? Asparta is synthesized by transfer of amino the ammonia group from glutamate to oxaloacetate. Asparagine is made by adding ammonia to aspartate. Let's see this one. Glutamate and oxaloacetate. These two will be compared into aspartate and alpha ketoglutarate, and aspartate will be converted into asparagine. See this one. This is oxidation, reduction, reaction. Now let's move to serine and glycine. Biosynthesis of serine and glycine start with oxidation of 3-phosphoglycerate and transamination with glutamate. This pathway with energy and form of reduced NADH. Let's see this one. 3-phosphoglyceric acid will convert into serine by three-step reaction. First is this one. This will be a reduction, and then glutamate will be converted into alpha ketoglutarate, and in the end it will be converted into serine. Then this serine by one-step reaction will convert into glycine. Now let's see the cysteine. Cysteine biosynthesis is a two-step reaction. Serine and acetyl-CoA combine to form O-acetylserine, and sulfur is then added to form a cysteine. Let's see this one. This serine, L-serine of course, convert into acetylserine, and by adding this sulfur, the acetylserine will be converted into cysteine. Now let's move to tyrosine. Tyrosine is formed from chorismate, the intermediate of many biosynthetic pathways convert from phosphoenol or pyruvate. Chorismate formation need one NADPH and one ATP. Let's see this one. Chorismate by a three-step reaction will convert into a tyrosine. Let's see this one. This is a reduction reaction. Then continue by converting this glutamate into alpha ketoglutarate. Then we'll get product of this reaction will be tyrosine. Now let's move to amino acid as a body fuel. Amino acid can be used as a source of energy, especially on starvation. Processing of amino acid result in metabolic intermediates such as pyruvate, acetyl-CoA, acetoacetyl-CoA, oxaloacetate, and alpha ketoglutarate. Let's see this one. We have a pyruvate, oxaloacetate, alpha ketoglutarate, succinyl-CoA, and fumarate. And we'll not forget about this one, acetyl-CoA. Now let's move to protein turnover. Balance between protein synthesis and degradation is required for good health. Protein turnover refers to the replacement of older protein as they are broken down within the cell. Different proteins have different turnover rate. Let's see this one, the figure in the right side. Let's say, for example, we take 100 grams of protein, and this protein will be digested to produce amino acid. And then this amino acid will be used or convert into a body protein. And a body protein can also convert into amino acid. Some of this body protein probably will digest, and the last story of this one will be secreted as a urea in our urine after filtration in the kidney organ. Let's see this one, 100 grams. Then protein breakdown is about 400 grams per day. So let's repeat it once again. The protein turnover refers to replacement of all the protein as they are broken down within the cell. Different proteins have different turnover rate. Now let's move to amino acid metabolism disorder, phenylketonuria. Phenylketonuria is an inborn error of metabolism that results in the metabolism of amino acid phenylalanine. This phenylketonuria caused by a mutation in a gene encoding phenylalanine hydroxylase. A sign for this amino acid disorder. Let's see this one. Odor from skin and urine, acne, seizure, tremor, and hyperactivity. That see this diagram or reaction in this slide. We have a phenylalanine here. This phenylalanine can be found from dairy protein or tissue protein. In a normal condition, this phenylalanine will convert into tyrosine. Then from tyrosine will convert into a several condition or a several compound, such as oxidation, protein synthesis, tyrosine, and melanin. But what happened with this one? As we have already mentioned that this is a kind of amino acid metabolism disorder. So this normal condition couldn't occur normally, then this will be switched off to others pathway and will produce others molecule. In this case, it will produce these two molecules. From phenyl pyruvate will produce phenylata and phenyl acetate. I think that's all for today or for this time. Thank you so much for your attention. If you have some or any question, just inform me or just write in the chat room in this channel. Again, thank you so much. See you then by next time, and thank you. Bye bye.